So here we have the life cycle of a DNA containing animal virus. So in this case, it's papovavirus. I wrote in the board some examples of important viruses according to the classification. So what we're looking right now, it's a non-envelope virus, a papovavirus. So this virus, in order to produce an infection in the animal cells, first it lands in the plasma membrane of a host cell, and with its receptors, it's going to attach, and then after it attaches, what it's going to do is going to penetrate. So this will be called entry and uncoating. So it's going to penetrate into the cytoplasm of the cell, and then all these capsid proteins that are part of the papovavirus are going to start disassemble. So this is the uncoating. So here you have all the capsid proteins, and this is the cytoplasm of the host cell, while this is the nucleus. And then the next step is going to actually integrate or bring this viral DNA into the nucleus. And once in the nucleus, a part of this viral DNA is going to be made into messenger RNA, which is going to be translated into early proteins. So you start exiting then this messenger RNA into the cytoplasm, and then you start making some early proteins, and then later on you start making the capsid proteins. Once you have these capsid proteins, that capsid proteins are going to go back into the nucleus, and then you're going to assemble a complete viral particle. And then that is the process of maturation. Once you have the particles assembled, and then you exit these particles through the plasma membrane of the cell. So then there is a question for you. Why the DNA from the virus moves into the nucleus? Why it doesn't stay in the cytoplasm, and why you replicate the DNA of the virus in the nucleus? Actually, what enzymes are used to replicate the DNA? DNA polymerases, right? So, where in the cell will be the DNA polymerases? In the cytoplasm or in the nucleus? The nucleus, right? That's where the DNA is. So often, with few exceptions, DNA viruses are replicated in the nucleus because that's where is the enzymes. So then, once you replicate your DNA, the capsid proteins that are made in the cytoplasm, then they will move into the nucleus. Where is the place of translation in the cell? Cytoplasm or nucleus, where you make proteins? In the cytoplasm. So then, what you expect is that you will have the making of the capsid proteins and early proteins in the cytoplasm, And in the case of a DNA virus, these capsid proteins are going to be imported into the nucleus so that you can assemble the viral particles in there. And then in general, with few exceptions as well, what you can expect then that RNA viruses replicate within the cytoplasm because that's where we have enzymes to handle RNA. And now here we have cycles of replication for RNA viruses. RNA viruses can be positive strand, negative strand. They can be single-stranded, double-stranded, and they can be segmented. If it's a positive strand, you will be reading that positive strand of RNA as if it was messenger RNA. Okay? And some viruses have that advantage. No matter which one it is, you will have this entry and then encoding into the cells. And then the viral genome, which is going to be RNA, is going to have different processes or undergo different processes depending whether it's positive or negative strand, if it's single-stranded or double-stranded. So for this case, we have a single-stranded RNA, positive strand, like in the case of picornaviruses. So once you have the encoding, you will have this positive strand translated into a negative strand, replicated into a negative strand, and also you will have the making of proteins out of this positive strand. And with this, you can make capsid proteins, and you will be able to replicate out of this negative strand this genome, the positive strand. And then you will have assemble and then release. For the negative strand, which is also known as antisense, like in the case of rhabdoviruses, once you have this viral genome encoded, so you will have this negative strand here, and out of this negative strand, you will make a positive strand to make proteins. And out of this positive strand, you will make more negative strands, and that is going to be important to make this viral RNA. And then with this messenger RNA, you will make your capsid proteins, and then you will assemble it, and then you will release. So that's for rhabdovirus. And now you can have this double-stranded RNA, which has a positive strand and a negative strand. But in this case, the machinery of our cells is incapable of replicating this DNA, this RNA. So what is going to happen is once you have your viral DNA inside this cytoplasm, you will have within this viral genome an actual RNA polymerase. And that RNA polymerase is going to be the one that is going to start making negative strands, and then out of these negative strands, you will make positive strands, and then you will have double-stranded RNA. So anytime that you have a double-stranded RNA, like in the case of reoviruses, you have to carry your own RNA polymerase because the one that we have is incapable of making double-stranded RNA. Okay, we only make, with our RNA polymerases, we can make only single-stranded RNA. So then once you have this double-stranded RNA, the rest of the RNA that you had here that you make it into messenger RNA, you can make these capsid proteins, and you can also make more enzyme, the RNA-dependent RNA polymerase, and then you will have everything complete to make a viral particle, and then you release it. So we will see this into a little bit more detail, but in general, expect that when the RNA is double-stranded, you have to have your own enzyme. Now, we have some viruses, like in the case of Retroviridae family, that they carry an enzyme as well, and these are RNA viruses, but this enzyme, it can make a copy of DNA out of RNA, and this is called reverse transcriptase, this enzyme that makes this. So we have here a host cell that receives actually this viral particle that it is from a retrovirus. In this case, it's an envelope virus, and it has these receptors that binds to the receptors of the host cell. And then this virus is introduced by endocytosis, and now inside the cytoplasm is going to uncoat, and it's going to release the viral RNA, and it's going to have its own enzyme, which is reverse transcriptase. So then, within this reverse transcriptase, it's going to use this viral RNA as a template, and it's going to make a double-stranded DNA. And this double-stranded DNA is going to be brought into the nucleus of the cell, the host cell, so that this viral DNA can integrate into here, forming a provirus, just like as what happened with the bacteriophage. That, like in the lysogenic cycle, the bacteriophage DNA integrated into the bacterial DNA. But in this case, this is an animal cell, like helper T cells, for instance, and then this viral DNA that it was made by this reverse transcriptase is integrated into the provirus. And this poses an advantage. So when the cells are infected and you show some antigens in the plasma membrane, the NK cells and the cytotoxic T cells can detect these viral infected cells and it will destroy them. But with this strategy, the viruses hide inside the cells. They don't show any of these antigens in the surface, and they will not be destroyed. So that's one way that the actual HIV seeds many cells in the body, and then when the person becomes immunocompromised, then this virus starts creating multiple copies, the cells will burst, and then the person will have like one of the stages of HIV infection, which is AIDS. So then, in order to replicate this virus out of this DNA that it was integrated here into the host cell, it will make messenger RNA. And then this messenger RNA will go into the cytoplasm, and it will be made into viral RNA, into viral proteins, and then you will have this assembly and then release. Now let's look at the differences in replication between animal viruses and bacterial viruses. So both of them have attachment stage. In the case of bacteriophages, they use the tail fibers to attach to the cell wall. In the case of the animal viruses, they use receptors to attach to receptors in the plasma membrane of the cells. Entry in bacteriophages, it happens by injecting the DNA into the cytoplasm, so there is no encoding, And in the case of the animal viruses, the entry can be through receptor-mediated endocytosis or by fusion of the envelope into the plasma membrane. And then what does the animal viruses require for uncoat? Well, they need some enzymes to remove the capsid proteins. And then biosynthesis for bacteriophages happens in the cytoplasm, and for animal viruses can be in the nucleus for DNA viruses or the cytoplasm for RNA viruses, with some exceptions. And then you can have chronic infection in both of them, which is called lysogeny in bacteriophages, and in the case of animal viruses, it will be called latency, slow viral infection, or sometimes some viral infections can lead to cancer. Release, it always happens by lysis in the bacteriophages, and in animal viruses can be through budding or rupture of the plasma membrane, which will be lysis. Okay, now let's look at characteristics of the viruses, including which can cause cancer. Also, we're going to look at the difference between persistent infections and latent viral infections. And we're going to also look at the specific viruses for other organisms like plants, and we're going to see prions and some viruses that infect humans. So in our cells, we have some genes in the DNA that are called oncogenes. And these oncogenes are capable of becoming activated and produce cancer. They can increase the transcription of genes within the vicinity, and these can be upstream or downstream genes, and that will lead to anomaly, especially when you have damage to genes that can affect the cell cycle. So we have some viruses that can integrate into the host cell DNA. And when they integrate this DNA into this area next to the oncogenes, these oncogenes can become activated and now are going to be called proto-oncogenes. And with these proto-oncogenes, our cells loses cell-cell contact inhibition. The mechanism, the molecular mechanism vary. Sometimes you can activate transcription of genes for replication. Other times you can do inactivation of tumor suppressor genes. And these tumor suppressor genes, which are housekeeping genes that we have that prevents the cells from replicating when they are not needed, then the cells will replicate over and over again. So you can have increased mitotic rate, or you can lose the cell-cell contact inhibition. For some viral infected cells, they can express tumor-specific transplantation antigens, or TSTA, or they can express T antigens in the nucleus. So these tumor-specific transplantation antigens is not that you are transplanting an organ. It is just a name that it was given to these type of antigens that sometimes can be expressed in the plasma membrane. But these antigens can be recognized by the immune system, and then the cells can start being recognized and being destroyed. And then for those that only express the antigens in the nucleus, well, in the nucleus we don't have surveillance by the immune system. So then that's how we miss the opportunity to detect these virally infected cells, and then we can have chronicity, and then depending on in which area of the genome this viral DNA is inserted is whether you will have a bad outcome or not. So here we have viruses that can cause cancer. oncogenes, oncogenic. Basically, what it means that they can produce cancer. So we have DNA viruses that are oncogenic, and then we have RNA viruses that are oncogenic. So examples of DNA viruses that are oncogenic can be adenoviridae, herpes viridae like Epstein-Barr virus, human herpes virus type 8, pox viridae, papoviridae, especially human papillomavirus 16, 18, and also 32, and then hepatnaviridae, which is hepatitis B virus. In the case of human papillomavirus 16 and 18, they inactivate tumor suppressor genes. So they inactivate, for instance, p53. And when this p53 is inactivated in both lines, the maternal parental line and the paternal line, then the person don't have these genes that prevents over replication of our cells. And then we have, for oncogenic RNA viruses, we have human immunodeficiency virus, we have human T lymphotrophic virus 1 and 2, or HTLV 1 and 2. We have flaviviridae, members like hepatitis C virus. Hepatitis C is one of these chronic infections in the liver with a virus that can actually lead to cancer in the liver. And it's a chronic infection. And then some can infect domestic animals like sarcoma viruses, and we have one specific for felines like feline leukemia virus. For some of them, we have vaccination. So actually, we have vaccination for this virus, and also for for hepatitis B and human papillomavirus. Here we have an example of an oncogenic virus causing lymphoma in the neck. This is called Burkitt's lymphoma, that it is produced by a virus. Now, for latent and persistent infections. So we have several viruses that can travel through the axons in nerves, like, for instance, the human herpes virus type 1 that causes cold sores. That one can travel, migrate through these nerves that we have in the facial area, and then specifically in the trigeminal nerve. It can go into the nucleus of the trigeminal nerve and live there for long periods of time. Initial infection, the person will have cold sores, which are pustules filled with viruses in the commissure of the lips. And then after this, you will have a period of remission. And when the person becomes immunocompromised, the person will have, again, the cold sores. And similar happens to shingles. It's out of the same family. It's a herpes zoster virus. So now this disease process occurs over a long period of time, and for some infections can be fatal, like in the case of measles virus, or even some viruses that they're not existing anymore, like this variola virus. So they were viruses that affects the entire brain tissue, causing inflammation of the brain, and then the severe inflammation leads to death in the case of persons. So when someone has this type of infections and happens in the brain, it's called subacute sclerosing panencephalitis. So here we have two cycles for what can happen with viral infections. You see this before, so this is the eclipse period. So here we have this graph showing you the number of virions and the number of days, months, or years for having a specific infection. Some of these infections, they can be acute, happens in the matter of days, and then you have a remission, and they can be found in the body for long periods of time, sitting viral particles little by little, and then you can have a sudden increase in numbers, and that leads to a latent infection. So here we have human herpes virus, and in this case will be chicken pox during childhood years, and then that can lead to shingles during adulthood. In this case, it's shingles in the thoracic area following the intercostal nerves. So these type of viruses, once they are in the body, they can have a retrograde migration through the axons in nerves, and then here you have the body of these nerves in ganglions that are found outside of the spinal cord, and in here you can have the viral DNA latent for years. Then when the person ages, and then the person becomes immunocompromised, or the person might be young and it's immunocompromised, these viruses start replicating again, and then they follow the pattern or the pathway of certain nerves, like in the case of the intercostal nerves, and then they can produce an infection, which in this case is shingles or herpes zoster. So this is a reactivation of a childhood infection. And this is what happens with cold sores. So you have cold sores in here, and then you have the nerves that makes the trigeminal. So these viruses can go into the ganglion of the trigeminal, and then when the person becomes immunocompromised, these viruses come back and produce another lesion. So you can have latency in nearby nerves. And this is how cold sores look like. So these lesions that are here are more or less dangerous than this, because these ones, they already dry out, so they don't have more viral particles, but these ones, they can break and spread throughout the skin in wounds. Questions? Okay, now for prions. So prions are proteinaceous infectious agents, and these prions can be acquired by inheritance. They can also be acquired by ingesting meat that is contaminated with these prions, by getting transplants contaminated with these prions, or if there are some instruments that are contaminated with these prions and the person has a surgery or a medical procedure, the person can have this infection. We have different names for these infections. In general, sometimes they're called spongiform encephalopathies, and they can be called chip scrapie, Creutzfeldt-Jacobs disease, Gerstmann, Strehler, Schenker, Macaulay-Schenker group, or fatal familial insomnia or mad cow disease. These were discovered a couple of decades ago, and it was kind of a surprise to think about that a protein can be infectious. And the protein that is infectious is called PRPSC, or scrapie protein. Now, in our brain, in our neurons, we have a protein that is similar to this scrapie protein that is called PRPC. That's the normal version of this scrapie protein. Now, when this PRPSC binds to the brain tissue, it gets internalized, and then it will convert these PRPSCs into scrapie proteins. And that is going to lead to precipitation of proteins within the neurons and cell death. and that will lead holes in the brain tissue that makes our brain tissue to look like if it was a sponge. That's why the general name for this disease is called spongiform encephalopathies. A mode of prevention, it is, of course, to make sure that the cattle or the animals that the humans consume, they don't have this scrapie. But when it comes to using instruments, surgical instruments, for instance, they have to be sterilized and they have to be pre-treated with either sodium hydroxide for 1 hour before sterilization, or they can also be treated with proteases, which will be able to break down these proteins. And then the autoclaving is going to be extended for longer time and at a higher temperature, which is 134 degrees Celsius. We know that the normal temperature or the type of temperature that we use, the level, it will be 121 degrees Celsius. So here is the cycle for infection for prions. So this is normal brain tissue with PRPCs, which are made, of course, by translation of messenger RNA to PRPCs. Then you put these PRPCs into the surface of the cells. And then if you have a PRPSC that comes and binds to these PRPCs, then these PRPCs will become PRPSCs. So they become prions. And that keeps going over and over again until you only have prions, you internalize them, and they precipitate inside the cells, and then you create holes in the brain. So these are the precipitated proteins as seen with the transmission electron micrograph in the brain tissue. And this is an autopsy of someone who died of spongiform encephalopathy. So these are holes in the brain tissue. And these are precipitated proteins. Questions about prions? All right. Now for plant viruses and viroids. Well, plant viruses are of different families, just as the animal cell viruses, and many of them are actually similar to the animal cell viruses, but they have different receptors that binds only to plant cells. And then plant viruses enters into plants using wounds or by insects binding into these plants. And these insects will have the virus in the proboscis that they use to bite these plants. Now, viroids are not viruses, but are only a short sequence of RNA that it is infectious and also can affect plants. So we have, for instance, potato spindle tuber disease, and here we have an example of this virus as seen with the transmission electron micrograph. So it's a short sequence of virus that can infect plants as well. So here we have virus families that infect plants. So we can have double-stranded, non-envelope, single-stranded RNA, positive strand, non-envelope. We can have single-stranded RNA, negative strand, envelope, and we can have double-stranded RNA, non-envelope. And here are the viral families. So here are members of these viruses and the mode of transmission. So we have here cauliflower mosaic virus that is icosahedral. It is medium in size, and it is transmitted into cauliflower plants by aphids. And then we have watermelon wilt virus that is a small virus that affects watermelon plants, and then it is transmitted by bites of these plants with whiteflies. And then we have virus, which is a spiral-like virus, is long, and this one infects tobacco plants entering through wounds in the plants. And then members of the Rhabdoviridae family, like the rhabdovirus that has the same family as this for animal cells. So we have potato yellow dwarf virus that same as for rabies virus. It has this bullet shape with this helical capsid, and of course it's an RNA virus, negative strand, and it enters into potato plants by the insects, aphids, and leafhoppers. And then we have a large size icosahedral virus, which is part of the Viroids family. It's a double-stranded RNA virus, non-envelope. This is called wound tumor virus and enters into many plants via leafhoppers, including grass. Okay, so those were the details for plant viruses. Any questions? Right, now for families that specifically infect humans. So I put you there at least the DNA viruses, but we're going to go over the RNA viruses as well. So the smallest DNA virus that we have are the human parvoviruses, B19, are part of this Parvoviridae family. They measure between 18 to 25 nanometers. It's single-stranded DNA and is non-enveloped. This one produces an infection that is called Fifth disease. And it is an infection that it can be easily recognized in little children because they have their cheek area very reddish. It looks like if they were being slapped. Okay, so it's actually colloquially known as baby or child slap syndrome. So it's by parvovirus B19, and again, it's the smallest of the DNA viruses. It's single-stranded DNA, non-enveloped. And now, of course, since you have now, for these two new viruses, double-stranded DNA, and this is non-enveloped viruses, you expect that the size become larger because you have more DNA, right? So we have adenoviruses that has spikes, and these ones measure between 70 to 80 nanometers. And then we have Papoviridae that they don't have the spikes as these adenoviridae and are a little bit smaller. So this one is the Papoviridae family that has human papillomavirus. And among some of these members of the human papillomavirus, we have the human wart viruses that causes warts in the skin, especially in areas of flexion like the knuckles, the knees, the elbows. And then we also have polyomavirus. So these viruses are known to cause warts and also cancer. Human papillomavirus 16, 18, 32 causes cervical cancer and also causes cancer in the anal area and as well in the larynx. Okay, so these viruses are the ones that I told you that inactivate P53, the tumor suppressor gene. And then for these viruses, we have vaccination. Now for adenoviruses, we have an important general like mass adenovirus. This one causes rhinitis or cold in us, and then in some animals causes tumors, like in the cows, can cause tumors in the udders. Now, for the double-stranded DNA viruses, well, in this case, you have an envelope, and these viruses, with the exception of the Hepatnaviridae, which is the smallest, of these ones, they are larger. So let's start with Hepatnaviridae. It is 4 nanometers in diameter, it's small, and the important family member will be hepatitis B virus. Hepatitis C virus, it is RNA virus. This one is DNA. And how do you know? Well, by the name of the family, and even for the name of the virus. Hepa for hepatic gland, which is the liver, and DNA, hepatna. So it's a DNA virus, the hepatitis B virus. And viridae, remember, is DNA in 4 families. So this one produces inflammation of the liver, so causes hepatitis. And this virus, it carries a reverse transcriptase to produce its DNA from messenger RNA. And this one, it is transmitted through contaminated blood transfusion, sexual intercourse that is unprotected, and parenteral route, like intravenous drug use with contaminated needles. And then we have vaccination available for hepatitis B infections. Now, for poxviridae, these ones are, again, double-stranded DNA viruses, and they are complex. They have an envelope. They measure between 200 to 350 nanometers, and we have important genera like vaccinia virus, smallpox viruses, and molluscopisovirus. So this one, it is a complex virus. It looks like a brick and causes a smallpox, which is also known as variola, and causes molluscum contagiosum, which is like a little dimple in the skin with a surrounding area that is a little bit raised on this dimple. And this is basically like a wart-like skin lesion, and it is also part of the family that causes cowpox infections. Herpes viridae, it's a large family. It has many members. They are medium-sized. They measure between 150 to 200 nanometers in diameter. Again, they are double-stranded DNA. They have this envelope. And then we have 4 simplex virus, type 1 and type 2. Type 1 infects mostly only the oral cavity area, and type 2 the genital area. In the oral cavity causes cold sores, and it causes pustules in the genital area. However, due to different sexual practices, then it has been found, because before it was only believed that type 1 infect only the oral cavity and type 2 only the genital area, but through different sexual practices, it has been found that actually type 1 can infect also the genital area and type 2 the oral cavity. And then type 3 is varicella virus. Type 4, lymphocryptovirus. Type 5 is cytomegalovirus. 6 and 7 are roseolovirus, and 8 is rhabdinovirus. So infections that they cause: chickenpox for varicella virus, fever and blisters for HHV1 and 2, shingles for type 2, infectious mononucleosis for 4, and then they can cause Burkitt's lymphoma in the case of type 8, the one that we saw with this big tumor in here in the neck before. So any questions about double-stranded envelope viruses? And then here we have smallpox, which we don't have anymore. This type of virus was eradicated few decades ago. However, this virus, it's, this virus actually it was eradicated because it used to create a lot of infections that killed people. This is roseola. It's what is called exanthem, enanthem, so basically it shows reddishness in patches in the skin without giving a raised area. This one is produced, this is infection produced by herpes virus type 6 and 7. And then 4 single-stranded RNA positive strand, so the ones that can directly be transcribed into, translated into proteins. So we have toga viruses that are medium size, between 60 to 70 nanometers, they are enveloped. And we have rubella virus, or rubivirus. So these viruses of these families of these toga virus can be transmitted by arthropods, like insects, and they can cause infections in the brain, like, for instance, Eastern equine encephalitis, Western equine encephalitis, and they can also be transmitted by respiratory route, like in the case of rubella. For this small virus, flavivirus, it measures between 40 to 50 nanometers in diameter, is the size of the Hepatnaviridae family. So this one has an important member, which is hepatitis C virus. This one causes hepatitis C. It's a chronic infection. We don't have vaccination. And then for other members, they can be causing infections like yellow fever, dengue, St. Louis, and West Nile encephalitis. And all of these ones are transmitted by bites of arthropods. Then we have coronaviruses that are very small, between 80 to 160 nanometers. And this one causes respiratory tract infections, and examples will be SARS virus and MERS-CoV virus. Then here we have an example of an infection by one of these viruses, rubella virus. Now for more negative-stranded RNA viruses, but in this case, some of them will have a complex shape like Flaviviridae, and all of them will be enveloped. So we have Rhabdoviridae that measures 70 nanometers in diameter by 100 nanometers in length, and we have a virus that is vesiculovirus that causes infections in the oral cavity, which are causing blisters. This is called stomatitis, vesicular stomatitis. And for rabies virus, which is part of the Lisa viruses, it can produce rabies in animals, or it produces. And then for flaviviruses or filoviruses, we have this, that it is very long. It can measure up to 14,000 nanometers in length. So we have the Ebola virus or Marburg viruses that have the same shape and they're long, and they cause hemorrhagic fevers because they infect these endothelial cells, and the endothelial cells are in the walls of the blood vessels. They rupture, and then you can have these hemorrhages. Then we have these paramyxoviridae, like measles virus. Measles virus causes measles, that it's an infection that affects mostly children, and it's been causing lately a lot of outbreaks. We have vaccination against this virus, and this one can cause encephalitis as well and death, but most commonly causes an exanthem and enanthem, similar to rubella. And then it can infect also chickens. It can cause Newcastle disease in chickens. And in these family members, we have also some viruses that causes cold diseases And then for Delta Viridae, this is the smallest virus from this family. It is 32 nanometers. And then we have hepatitis D. And hepatitis D is an effective virus. It needs the envelope from hepatitis B to cause an infection. So you have to have co-infection with hepatitis B and hepatitis D to have hepatitis D infection. We have here a case of mumps. So this is a virus that can infect the parotid gland. For single-stranded RNA, positive strand, non-envelope, we have Picornaviridae, which is the smallest RNA virus that we have, measured between 25 to 30 nanometers. And we have several important members like enteroviruses that causes diarrhea, rhinoviruses that causes common cold, and hepatitis A virus that causes hepatitis A. Now, since these viruses that are naked viruses, they don't have an envelope, they are more resistant to the environment, something that you expect is that these viruses are transmitted via fecal oral route. The rest that are enveloped, that are more susceptible to the environment, they are transmitted through nasal secretions, through body fluids, etc. So then we have around 100 rhinoviruses, and we have seasonal viruses that, just like as the influenza virus, can cause infections in certain periods of time during the year. And then we also have an important member here, which is poliovirus, which is an enterovirus. Poliovirus causes infections in the nervous system, specifically infects the motor nerves, the spinal cord, and that can lead to the person having lack of innervation by these nerves and then atrophy of the limbs. And for poliovirus, we also have vaccination. And then for Calliciviridae, we have hepatitis E virus and noroviruses. These are transmitted fecal orally, and in the case of hepatitis E, we don't have vaccination, but it is just as it is transmitted just as hepatitis A, and is through the fecal oral route, and it has similar period of infection, just as hepatitis A. Now, for negative strand viruses that has an envelope, we have this small size virus, Orthomyxoviridae, it has this envelope. It has segmented RNA. We have like 7 segments, and we have important members like influenza virus A, B, and C. This virus can infect not only humans, but also can infect birds and swine. And when there is close contact between humans and these animals, there is transmission of these viruses between species, and the viruses can interact, and then you can have truffling of pieces of RNA between the viruses, and then you create a new strain. And that's how we end up with pandemics that are very important with these viruses. And then these viruses have enzymes that can agglutinate the red blood cells, like neuraminidase. And then we have this medium-sized virus, like Bunja virus, that measures between 90 to 120 nanometers. We have California encephalitis virus and hantavirus. This hantavirus causes hemorrhagic fevers and causes hantavirus pulmonary syndrome. This hantavirus has a vector, rodents. Rodents can carry the virus in its body, and when they are in areas like, for instance, supermarkets, and where they keep all these canned items, these rodents can pee and poo there. They can leave their droppings. And then if someone consumes an item out of these cans, they can get the hantavirus and then they can get the infection. So what you hope for is that they keep all these items for human consumption out of the pathway of these animals, and hopefully they don't have these animals around with these infections. But it is important then, whenever you get a can of food or soda, clean the top really well to prevent maybe having some of these infectious diseases. And this is not the only one, but this is an example of how this virus can go into your body. And then for arenaviridae, it has segments of RNA that makes it look like if it was particles of sand. That's why it's called arenaviridae. Arena means sand. It measures between 110 and 130 nanometers in diameter. It has a helical capsid, and it can produce infections in the brain like lymphocytic choriomeningitis or Venezuelan hemorrhagic fever and Lassa fever. For Retroviridae family, we have this HIV, which measure between 100 and 120 nanometers. This virus infects the lymphocytes type 4 or the helper T cells, and as well infects macrophages and dendritic cells in the brain. Then we have rotaviruses, which are part of the Reoviridae family. They are very small, double-stranded RNA non-enveloped viruses. They measure between 60 to 80 nanometers in diameter and mainly causes mild respiratory infections. And as well, they can cause infections in the gastrointestinal tract. So lastly, for hepatitis viruses, we have this classification with letters. And these are the first 5 that we're going to see. There are a few more, but these are like the most common ones. So we have A, B, C, and D and E. A and E are transmitted fecally orally, and the rest are transmitted by the B and C by sexual intercourse, unprotected sexual intercourse, parentally by intravenous injection of drugs with contaminated needles, blood transfusions, or receiving a transplant that is contaminated with these viruses. So the incubation period, which is the time that it takes for you to get in contact with the virus and getting the first signs and symptoms. So you have between 2 to 6 weeks for both hepatitis A and E. Fecal oral route, mode of transmission for both. And then diagnosis test will be to measure the levels of antibodies, which will be IgM for both. And for hepatitis, both A, E, and A, you can use PCR. And actually for each of these, you can use PCR. And then treatment is to provide antibodies for hepatitis A, none for hepatitis E. And this one can be prevented by having vaccination with inactive viruses or by giving antibodies if you get exposed to this. And apparently for hepatitis E, hepatitis A vaccination is protective. Now, for hepatitis B, the incubation period is similar to hepatitis D because just in case that someone has hepatitis B and get in contact with hepatitis D at the same time, the person can get a co-infection. So it's between 4 to 26 weeks. So that's how long it takes for the person to get signs and symptoms. The signs and symptoms of any hepatitis initially, it is fever, cephalia or headache, myalgias, arthralgia, so pain in your articulations, in your muscles, and also having a yellowish tint in your skin. So that would be ictericia. And then I told you already the mode of transmission for hepatitis B and C. For the incubation period for hepatitis C is shorter than hepatitis B, between 2 to 22 weeks. And then vaccination only for hepatitis B we have. And then the treatment will be giving for hepatitis B and C interferons and nucleoside analogs for hepatitis B. We will see that in the antiviral agents chapter about which ones are nucleoside analogs. And then for hepatitis C, you give interferons and ribavirin. Sometimes people with hepatitis C can get cured with these interferons. Any questions?